Sebentar ya. Okay, good morning everyone. Good morning, Ma'am Retno. Today, me, Rahul, and Mazaya from Group D will present about our topic, which is amino acids and proteins. Next.
So these are the members of our group. And the journal that we chose is the analysis of protein amino acids, non-protein amino acids, and metabolites, dietary protein, glucose, fructose, sucrose, phenolic, and flavonoid content, and antioxidative properties of potato tubers, peels, and cortexes pulps. So the summary or analysis of this article is this study. 16 freeze-dried powders were analyzed for protein and free amino acid content. And the highest protein powders may add to dietary protein, but their nutritional quality must be assessed. The variation in asparagine content of whole potatoes suggests that, like other potato cultivars, some Korean cultivars have low asparagine content, and it results in low acrylamide levels after frying or baking. And while the search for asparagine-free potatoes continues, a major development is that silencing the asparagine synthetase enzyme lowers both asparagine production in raw potatoes and the acrylamide forming potential of processed potato products.
Okay, so I'm going to start with the first subtopic, which is the chemical synthesis of peptides. So peptides are used to develop etiopate specific antibodies that map antibodies, etiopes, and enzymes binding sites and develop new enzymes, medicines, and vaccines. Peptide synthesis used to be a labor-intensive and yielded low yields, but new manufacturing methods in peptide chemistry have been made it more efficient, more readily available for common research purposes. There are two sorts of peptide synthesizers. The first one is the liquid phase synthesis, which is a classic approach to peptide synthesis, is a liquid phase peptide synthesis. Solid phase synthesis has largely superseded in most labs. However, it is still useful in large-scale peptide manufacturing for industrial uses. The second synthesis is the solid phase synthesis, which enables the synthesis of natural peptides that are difficult to express in bacteria, as well as the integration of non-natural amino acids, peptides/protein backbone modification, and the synthesis of D proteins. So there are 3 steps or 4 steps. There are 3 steps for the SPPS or solid synthesis. The first step is peptide deprotection. Because amino acids contain various reactive groups, peptide synthesis must be done with care to avoid side reactions that shorten the peptide chain and cause branching. Chemical groups that attach to the reactive groups of amino acids and block or shield the functional group from non-specific reactivity have been created to enable peptide synthesis with minimal side reactions. It is activated in the next phase after being deprotected. You can see in the diagram on the right-hand side that the first step is the deprotection, then activation. So the second step is the amino acid coupling. The synthetic peptide coupling requires the activation of the C-terminal carboxylic acid on the incoming amino acid and using carbodiamides such as dicyclohexocarbodamide or DCC in short. The third step, as you can see from the diagram, the coupling method and the coupling step, then it goes to the peptide cleavage. All residual protecting groups in the natural peptide must be eliminated after the consecutive cycles of amino acid deprotection and coupling. Strong acids like hydrogen fluoride, hydrogen bromide, or trifluoromethane sulfonic acid, or TFMSI, are used to cleave BOC and BZL groups, while TFA is used to cleave MFOC and TBUT groups. Cleavage removes the N-terminal protective groups of the final amino acid supply. The C-terminal protecting group either chemical or resident from the initial amino acid and any side chain protecting groups when done correctly. So the next subtopic, the next part is the biosynthesis of the peptides. So this information teaches a cell how to make the proteins it needs to survive is encoded in the DNA structure in its nucleus. Because there are only 4 nucleotides and 20 amino acids to code, this code cannot be based on a one-to-one match between nucleotides and amino acids. There are 64 potential triplets or codons if the nucleotides are arranged in 3, which is more than enough combinations to code for 20 amino acids. We must break down the decoding process into 2 parts to understand how proteins are created. The first step is the transcription. A little piece of DNA double helix must be uncoiled before the information in the DNA can be deciphered. A complementary copy of one strand of DNA is then produced as an RNA strand. Assume that the copy piece of DNA has the following nucleotide sequence, starting at 3N. So I can see from the figure below the transcription, there is this sequence. And when predicting the nucleotide sequence in RNA complement, keep in mind that RNA employs U instead of T, as in the DNA. We must also keep in mind that the base pairing happens when 2 chains run in opposite direction. As a result, the RNA complement of this DNA should be written as follows. As you can see from this figure, is a mRNA sequence and the DNA sequence, and you can see there is a difference. Next. So the second step is translation, where the messenger RNA now connects to the ribosome, which converts the message into an amino acid sequence. The amino acids that are integrated into the protein being made are carried by the transfer RNA or tRNA, or transfer RNA in short, which are relatively small RNA molecules. The relationship between the triplets or codons on the mRNA and the amino acids is shown in the table below. As you can see from the table, specific nucleotides are used to create specific amino acids. You can see tyrine, or you can see ruin, you can see many amino acids. Next. So the signal to start a polypeptide chain in simple prokaryotic cells is the triplet AUG, which codes for the amino acid methanoid, or MET. The synthesis of every protein in the cells therefore starts with the MET residue at the N-terminal of the polypeptide chain. A tRNA carrying the second amino acid attaches to the next codon, and after the tRNA carrying the MET connects to the messenger RNA start signal. When the MET residue on the first tRNA is transferred to the amino acid on the second tRNA, a dipeptide is formed. The dipeptide would be MET-PHE, is reading from the N-terminal to the C-terminal of the amino acid. If the DNA described in the section was translated, a tRNA containing the third amino acid, VAL, attaches to the next codon when the messenger RNA proceeds through the ribosome. As you can see from the sequence, we see here MET-PHE, VAL, ASN, GLN, and HIST. This polypeptide is not necessarily an active protein. All proteins in prokaryotic cells start with MET when synthesized, but not all proteins have MET first in their active form. It is often necessary to clip off this MET after the polypeptide has been synthesized to give a protein with a different N-terminal amino acid. The protein classification will be explained by Regina.
Okay, so next we have the protein classification. Next slide. So firstly, we have the protein classification based on their chemical composition. So basically, the first one, we have simple proteins, also called homoproteins, which are comprised entirely of amino acids. Plasma albumin, collagen, and keratin are some examples. And next, we have conjugated proteins, which are also known as heteroproteins. And they have a non-protein part in their structure. So glycoproteins, chromoproteins and phosphoroproteins are 3 examples of this. So for glycoproteins, they are proteins that are attached to one or more carbohydrates unit to the polypeptide backbone in the covalent manner. And chromoproteins are proteins that contain colored prosthetic groups. So for example, phosphoproteins are also the proteins that bind phosphoric acid to serine and threonine residues. Next. And for the second one, we have the protein classification based on shape. So first we have fibrous proteins, which are essentially mechanical and structural proteins that provide support to cells and the entire organism because they contain many hydrophobic amino acids internally and on their surface. These proteins are water insoluble. And hydrophobic amino acids on the surface help their packing into exceedingly complex supramolecular structures. Fibrin, collagen, keratins, and elastins are examples of this. And we also have globular proteins, which almost all proteins are classified as globular. They have more complex structures than fibrous proteins, which are compact and more or less spherical in shape. And in addition to secondary structures, motifs, domains, tertiary, and quaternary structures are discovered, and this will be explained more on the next slides. And they are also soluble in water in general, but they can also be found integrated within biological membranes, such as transmembrane proteins, which put them in a hydrophobic environment. And Next, we have protein classification based on their biological functions. So the presence of many different functional groups in the amino acid side chains, such as thiols, alcohols, thioethers, carboxamides, carboxylic acids, and different basic groups, as well as the folding of the polypeptide chain. This will result in their 3-dimensional structure, which allows the protein to provide a wide range of functions. For example, they can act as enzymes, which are biochemical catalysts. Enzymes are specialized proteins that catalyze practically all the processes in living organisms. They have high catalytic power and they raise the rate of reaction in the reactions that they are in by at least 106 times. As a result, life as we know it would be impossible without these biochemical catalysts. And almost all known enzymes are we have thousands of them in our human bodies, and they are proteins all except for the catalytic RNA molecules, which are ribosomes. And next, we also have transport proteins, which are specific protein transports that transport many tiny molecules, both organic and inorganic, and in the circulation and extracellular fluids across cell membranes and inside cells from one compartment to another. Next. Okay, now we are going to discuss the protein structures, and this includes primary, secondary, tertiary, and quaternary. Next, Okay, so firstly, for the primary structure, the simple definition is that it is the sequence of amino acids that are linked together to form a polypeptide chain. And basically what amino acids are, they are small organic molecules that consist of a chiral carbon with 4 substituents, and the fourth site is different amongst each of the different amino acids. So the sequence that they are in are determined by our DNA, and the amino acids are linked to the neighboring amino acid through peptide bonds, which are formed in the biosynthesis process. And here in the biosynthesis process, a water molecule is extracted and it joins an amino group with a carboxyl group of a neighboring amino acid. And the covalent peptide bonds maintain the primary structure of the protein sequence. Next. So for secondary structures, the simple definition is that they are folded structures that form within a polypeptide due to interactions between the atoms of the chain. Here in the secondary structures, the R groups are not included, and they are commonly folded into 2 stable configurations consisting of alpha helices and beta pleated sheets. Next. So for alpha helices, they fold into a coil/spiral arrangement seen in the figure in the slide, and the carbonyl or the CO bond is the amino acid is bonded to the NH group that is four carbons down the chain, and this bonding pattern pulls the chain into a helical shape. And the R groups also stick outwards from the helix, and they are free to interact with the compounds of the environment. Next. So for beta pleated sheets, this is a little bit more complicated. They fold into a directionally oriented staggered strand configuration seen in the figure below, and more or equal to two segments of the polypeptide chains line up next to each other, forming a sheet-like structure with hydrogen bonds, and the hydrogen bonds form between the carbonyl groups and the amino groups, and they exclude the R groups like the alpha helices, and they extend above and below the sheet plane. These strands can also be parallel or anti-parallel. If they are parallel, the points it points in the same direction, hence the N and C terminus aligns. And if they are anti-parallel, it points in opposite direction. Hence, the N terminus of the strand is next to the C terminus of the neighbor. Next. Okay, for tertiary structures, they are the overall dimension structure of the polypeptide due to the interaction between the R groups. So the definition is quite similar to the secondary structure. The difference is that they include the R groups of the proteins. So for the R groups that affect this, they are all non-covalent bonds, which can be hydrogen bonding, ionic bonding, the dipole-dipole moment, or the London dispersion forces. And hydrophobic interactions can also affect the structure. And we can see this when amino acids with non-polar hydrophobic R groups cluster onto the inside of the protein and leave the hydrophilic amino acids facing the outside to allow them to interact with the surrounding water. And lastly, disulfide bonds or covalent linkages can also affect this. Between the sulfur-containing side chains of cysteines, which are much more stronger than other bonds, and they basically act like safety pins. Next. Okay, so for the last protein structure, it is the quaternary structure. The definition of this is that it is the structure of a protein macromolecule, which are formed by interactions between multiple polypeptide chains. And each polypeptide chain is referred to as a subunit. And the proteins with a quaternary structure may consist of more than one of the same type of protein subunits, and they may also be composed of different subunits. So hemoglobin is an example of this, and it is found in the blood. It is found in the blood and is an iron-containing protein that binds oxygen molecules. It contains 4 subunits, which are 2 alpha subunits and 2 beta subunits. Next.
Okay, so the last subtopic is protein denaturation. So protein denaturation's definition is the destruction of the tertiary structure of a protein molecule and the formation of a random polypeptide chains. And this results in the disturbance of stability and structure of the protein. One example of protein denaturation is the coagulation of egg white when an egg is subjected to boiling. And in here, the denaturation occurs due to the change in temperature, and the figure for protein denaturation is shown on the screen. So for the causes of protein denaturation is first because of temperature. So high levels of thermal energy may disrupt the hydrogen bonds that hold the protein together, and as these bonds are broken, the protein will begin to unfold and lose its capacity to function as intended. And most human proteins function optimally at body temperature. So the second cause is pH. So amino acids are deuterions, neutral molecules possessing both negatively COO- and positively NH3+ charged regions. And changing the pH of a protein will alter the charge of the protein and in turn will alter protein solubility and overall shape. And all proteins have an optimal pH, which is dependent on the environment in which it functions. So for the denaturations at levels of protein structure and primary structure, such as the sequence of amino acids held together by covalent peptide bonds is not disrupted by denaturations. For secondary structure denaturations, the proteins lose all regular repeating patterns such as alpha helices and beta pleated sheets, and it will adopt a random coil configuration. For tertiary structure, the denaturation involves the disruption of first is the covalent interactions between amino acid side chains, such as disulfide bridges between cysteine groups, and also non-covalent dipole-dipole interactions between polar amino acid side chains and the surrounding solvent, and also van der Waals-induced dipole interactions between non-polar amino acid side chains. And in quaternary structure denaturations, the protein subunits are dissociated and or the spatial arrangement of protein subunits is disrupted.
Okay, so that is all for our presentation, and these are our references. Thank you for listening to today's presentation.